We are measuring biodiversity, and we are specifically going to be using Simpson's diversity index, but we also know there are many other ways to calculate some kind of biodiversity index. Our specifics is Simpson's diversity index. You will see many others, and you would expect that all of these types of measurements you're going to need some kind of fieldwork samples to have been collected, quadrat images, whatever it is. You might just have raw counts of specific organisms, or you might just have different species A, species B. You can differentiate between them, but you don't necessarily know what they are. That is still perfectly fine. So let's look at some field data from two particular areas of grassland. We've got some flower species happening, area 1, area 2. They both have 3 species in them, and our measurements of diversity have been these particular ones. Now we could easily calculate richness and evenness because we have raw data. In order to do percentage cover and percentage frequency, we would need some more high quality quadrat data, some pictures, whatever it is. We can't use the numbers to do those ones, but we can definitely combine these two to calculate SDI. So let's do species richness first. Remember, we're using Mennenich's index here, and it includes the number of different species and the total number of individuals of all species. So super simple, really. We're just doing some substitution. Here's our formula. We're putting in 3 because there are 3 species and there are 1,000 organisms here in this area, and our answer here is this. We're not going to use units here. All we're going to do is compare the numbers from area 1 to area 2, pardon. So same thing here. In area 2 there are 3 species, also 1,000 total, so we're going to end up with the same substitution and therefore the same number. So this actually doesn't really tell us much. We can see immediately there are big differences in diversity. We can see this one is dominating, but this number, as the only measure we've done so far, is kind of telling us that these areas are exactly the same in terms of biodiversity, which we can see is not the case. So we are going to consider evenness or relative abundance here in this situation, and the way we can do that is by calculating some percentages, and we can see the percentage of daisies, dandelions and buttercups is quite drastically different between area 1 and area 2. So we can say that area 1 with its 30, 33 and 36 percentage as opposed to 2, nearly 5 and 93, that area 1 is more evenly spread, and this has very different distribution to area 1. Now, if we consider both the richness and the abundance, or the evenness, we can calculate SDI. And remembering that, we've got a little n here. We're going to have this is our sum. Our sigma is our sum of. The little n is the number of individuals in each species, so like 300. And n take 1 is just that number, 300 take 1. And our capital N is the total number of organisms of all species, so there's that 1,000. So if we start substituting that in, we can see we've still got to keep the 1 subtract. We've got 3 different, because we have to do the sum of all 3 of these species. So 300 times 300 take 1. We've got 335 times 335 take 1. Same thing here, 365 times 365 take 1. It's not a complicated formula. It looks complicated, but it could get quite long if you have many, many species. We've got our total on the bottom, 1,000, 1,000 times 1,000 take 1. That's it. We come up with a number which we are subtracting from 1, and we get this number here. Once again, we are aiming for between 0 and 1, 1 being infinite biodiversity. We can see this is, past the halfway point, but really what we want to do is compare area 1 and area 2 is a relative measure here. Same thing, substitute in, we've got a 20 times 19, a 49 times 48, and a 931 times 930. Our totals are the same, so same substitution there in the denominator, and we will end up with a smaller number. So the way we can interpret that, firstly, very broadly, is to say this has higher diversity. Area 1 has higher diversity than area 2. Another way we can interpret that, however, because SDI is talking as percentages, we could interpret this by saying that area 1, if it's sampled and we chose 2 individual organisms, there's a 67% chance that they're going to be from a different species. Or, alternatively, you can say that they are going to be, we'll do the reciprocal there, there's a 33% chance that they are from the same species, right? So that's not a huge, it's not a huge number, which means actually there's quite a lot of biodiversity there. Similarly, with area 2, you can say there's going to be a 13% chance that if I take 2 random organisms, they're actually going to be from different species, which means, alternatively, the reciprocal of that is 87% chance they're going to be from the same species. And we can see, yeah, there's probably going to be a pretty high percentage chance that I picked 2 buttercups right there. So another way this can be organised using field data is in a table of values. And we can look at this list of species in a similar way to looking at a frequency table. To find the rest of the values of our SDI calculation, we can just put new columns in. So we need an n column, an n-1, and an n times n-1 column. And this is really handy if you have so many species that it's just not going to be very viable to substitute it all into the formula. So here's our n-1 column, super simple if we're just looking at our nice raw numbers here. And then same thing when we have an n times n-1 column. We simply have to go this column times that column, what's our answer? And at the end, we have all of our totals. So when we're looking at our formula, we've got sigma or sum of n times n-1, we've already got that number available. So when we substitute in, we just have to put in 2 particular numbers. So if we keep going with that, we've got our 64 multiplied by, we know our total is 15 times 14, or the capital N times capital N-1, and our value is going to be 0.3 here. So remember that there is a couple of different interpretations of what this means. If we're only looking at one individual area, we can't necessarily say it's very high or very low compared to anything. We know it's closer to the lower side because it's closer to 0, but we could also interpret that, sorry, this is actually incorrect. We can interpret that by saying 30% chance that the 2 organisms are from a different species, so this is incorrect, or there's a 70% chance they're from the same species. And we can see we've got holly here, which is dominating the area, and that makes total sense. I invite you to work through this particular problem by looking at and doing a basic count of the species and then pausing this and coming back and looking at your answer. Species richness, species evenness, and species diversity for community A and community B, and making some comparative statements at the end there. You can see we don't know what kinds of species they are, so you could maybe call this one A, and we could have to identify it over here to say this is A as well, and go from there. And I invite you to come back. I'm going to put all the answers on the screen, so maybe do it after you've paused this and come back and check your answers, because there's quite a lot. So we've aimed to determine the diversity of species using a whole lot of these. We have actually given examples of species richness and evenness in this particular video, but our focus really is doing various ways to find SDI. You might just substitute into this formula, making sure you understand all elements of it, or you are organizing a table and then substituting in, depending on what your preference is.